Let us discuss the comments in your practical metabolism part. You have analysis of SGOT and SGPT in blood serum. What is SGOT and SGPT? Serum glutamic oxaloacetic transaminase, that is SGOT. It is also called as aspartate aminotransferase, AST, and the other one is serum glutamic pyruvic transaminase or alanine aminotransferase. These are the two enzymes involved in exchange of amino groups and oxa groups between alpha amino acids and alpha oxo acids. This reaction is an example that is catalyzed by SGOT. Here aspartate reacts with alpha ketoglutarate to form oxaloacetate and glutamate. Here vitamin B6, that is pyridoxal phosphate, acts as a cofactor in the reaction. Here the transfer of amino and oxo groups occurs in the reaction. It is catalyzed by SGOT, that is serum glutamic oxaloacetic transaminase. So what is the importance of SGOT in our body means elevated levels of SGOT are usually observed in any damage or disease of heart and liver. So the lesser amount of SGOT are found in skeletal muscles. When any damage for your heart and liver is recorded. Then in kidney, pancreas, spleen, lungs and brain damage to these tissues also results in slightly rise in SGOT levels. Here you can see the graph in case of acute myocardial infarction. Once the pain is onset, after 6 to 8 hours, SGOT appears. After 18 to 24 hours, you can see the peak level of SGOT. Later, it will come to normal level after 4 to 5 days. So let's know about SGPT. So SGPT is also called as ALT, that is alanine aminotransferase. Here you can see in the reaction L-alanine reacts with alpha oxoglutarate to form pyruvate and L-glutamate. This reaction is catalyzed by SGPT. As I have told you, SGOT and SGPT both helps in the exchange of amino and oxo group between the alpha amino acids and alpha oxo acids. This graph shows the level of SGPT in case of jaundice or viral hepatitis. So SGPT rises once the jaundice arrives, then it will go to peak level in around 7 to 12 days. Again it will come to normal level after 3 to 4 weeks of the jaundice. So this is about the importance of SGOT and SGPT in blood serum. So the elevated levels of SGPT are usually observed in hepatitis. You are seeing in the picture, then the muscle disease and traumas also. It is mainly used in diagnosis of liver damage. Then it can also be used in combination with SGOT for diagnosis of myocardial diseases. Next, you have phenylketonuria. So, what is phenylketonuria? Phenylketonuria comes under the inborn errors of metabolism. We say inborn errors of metabolism, they are a large group of rare genetic diseases. Okay, they generally result from a defect in an enzyme or transport protein, which results in a blockage or block in a metabolic pathway. So the effects are maybe due to the toxic accumulations of substrates before the blockage or intermediates from alternative metabolic pathways. So the defects in energy production and also use caused by a deficiency of products beyond the blockage. So the metabolic deviation can also often affects the central nervous system, leading to some neurological disorders. So phenylketonuria is also an rare inborn error in which metabolism that causes accumulation of amino acid called phenylalanine in the body. In this case, you can see the accumulation of phenylalanine in the body. This is due to the defect in the gene encoding for phenylalanine hydroxylase. Okay, phenylalanine hydroxylase is an enzyme that converts phenylalanine into tyrosine. You can see in the reaction. So tyrosine is essential for the synthesis of neurotransmitters such as epinephrine, norepinephrine and dopamine. So the untreated So here you can see in the presence of the gene encoding for phenylalanine hydroxylase, the reaction will be the phenylalanine is converted into tyrosine by the reaction catalyzed by phenylalanine hydroxylase. So green one is a phenylalanine hydroxylase gene. In the presence of the gene, everything will be normal. In this case, there will be a lack in phenylalanine hydroxylase gene. So the phenylalanine hydroxylase will be non-functional. Hence, the phenylalanine will not be converted into tyrosine. This further leads to complications. So as I've already told you, tyrosine, so formed from phenylalanine, is required for the synthesis of neurotransmitters such as epinephrine, dopamine, norepinephrine, so if the level of tyrosine is less, it leads to abnormal brain development and intellectual disability. Dopamine and norepinephrine are also needed for serotonin. Because dopamine and norepinephrine are synthesized by tyrosine, if phenylalanine is not converted to tyrosine, hence the reaction stops leading to phenylketonuria. So how it can be treated? we should follow, one should follow low phenylalanine diet and no phenylalanine formula, synthetic form of protein, and high protein foods must be eliminated from the diet. Each patient will need to consume the amount of phenylalanine dietary intake. Untreated phenylketonuria can lead to intellectual disabilities, behavior problems, seizures, and mental disorders. It may also result in a mousy odor, that is musty smell, and lighter skin, that is hypopigmentation, eczema. So we need to increase the tyrosine in diet to overcome it. Then phenylketonuria is also shows a symptom, that is phenylketones will be present in the urine. It is an autosomal recessive disorder. Then we have another inborn error of metabolism, that is hypercholesterolemia. Hypercholesterolemia, by the word, it is excess levels of cholesterol. Okay, it is a familial disorder. Familial means runs in the family. It is a genetic disorder, you can say. Hyper means excess, then cholesterolemia means levels of cholesterol in blood. So cholesterol that is present in cell membranes acts as the precursor to steroid hormones, bile acids, and vitamin D. So hypercholesterolemia is an elevation of total cholesterol, or LDL, that is low density lipoprotein, then non-high density lipoprotein, that is HDL. It is also defined as the subtraction of HDL from total cholesterol in the blood. It is also referred as dilipidemia. In cholesterol we have LDL and HDL, two different forms of cholesterol, that is low density lipoprotein and high density lipoprotein. High density lipoprotein is a good cholesterol, we say, and low density lipoprotein is a bad cholesterol. So high density lipoproteins helps to remove the excess cholesterol in your body through urine. Low density lipoprotein is a bad cholesterol. It deposits on the cell membranes and also results in the atherosclerotic plaques. So what are the symptoms of hypercholesterolemia? Means it is related to xanthomas, tendon xanthomas, tendons of hands, elbows and knees. Then it is also called as Achilles tendon. Another form is called xanthelasma palpebrarum, that is the formation of yellow patches of cholesterol, that is deposition of cholesterol that looks like yellow patches in the eyelids. It also leads to coronary arteries, cerebral arteries, heart attack and stroke. What is the treatment for this? We have to change the lifestyle. We need to avoid junk foods, smoking, etc. Statins are the drugs to treat hypercholesterolemia. Next is gluconeogenesis. Gluconeogenesis is the synthesis of glucose from non-sugar precursors such as lactate, pyruvate, and the carbon skeleton of glucogenic amino acids. Here you can see production of glucose from pyruvate in order to maintain steady blood glucose levels. Gluconeogenesis is stimulated by the diabetogenic hormones such as glucagon, growth hormone, epinephrine, and cortisol. Glucogenic substrates include glycerol, lactate, propionate, and certain amino acids. When we fast or when we are in fasting, the body maintains glucose level in the body using gluconeogenesis process. How? Means, so we can see the metabolic pathway here to make glucose from amino acids, lactate, and glycerol. Primarily in liver cells, it occurs also in the epithelial cells of the kidney and intestine. Urea cycle. Urea cycle is a sequence of 6 enzymatic and 2 transport steps necessary to metabolize and excrete the nitrogen or ammonia generated by the breakdown of amino acids in protein and other nitrogen containing molecules in the body, except aspartate. So the ammonia produced in the body from all other amino acids and nitrogen containing compounds is to be excreted. So this is done in urea cycle. Ammonia, the toxic ammonia is converted into urea and excreted out of the body through urine. In this reaction, this cycle is also called as ornithine cycle. Here, ornithine is an intermediate compound formed in the cycle. It acts as a chemical scaffold on which a moiety of urea is built. Then the finished moiety of urea is discharged by hydrolysis process, leaving free urea and also regenerating the original molecule of ornithine for the next cycle. So mainly it converts ammonia into urea. See in the picture you can see ornithine releasing urea and it will move on to next cycle. It acts as a chemical scaffold.